Hello and welcome to my tutorial on the electron transport chain. So we're going to first talk about what the electron transport chain is. It's a series of carriers and pumps that releases energy in the form of ATP. These are found in the inner mitochondrial membranes, or if you are a plant, you will find it in the thylakoid membrane of the chloroplast. So I'll draw a cross section of a chloroplast. So where all the action happens is on it's all going on here in the thylakoid membrane. So yeah, and the mitochondrial membrane is right here. So it will take place in the intermembrane space, the sort of peachy part of my diagram here. And so yeah, we'll carry on. CO2 is a waste product of respiration, but the hydrogen atoms are a potential source of energy. Hydrogen atoms are carried by the coenzymes NAD and FAD into the electron transport chain. If produced, NAD is the initial acceptor. For each pair of hydrogen atoms involved, enough energy is released with the synthesis of 3 molecules of ATP. If reduced, FAD replaces produced NAD as the first carrier. Only 2 molecules of ATP are produced. The most widely accepted explanation of ATP synthesis in oxidative phosphorylation is chemiosmotic theory. So chemiosmotic theory is a model to explain the synthesis of ATP. The theory proposes that the energy for ATP synthesis originates from the electrochemical gradient of protons across a membrane. An oxidative phosphorylation occurs in the membranes of the mitochondria during aerobic respiration, and a coenzyme is a molecule required by some enzymes in order to function, which is what NAD and FAD are. They are coenzymes. So we'll now move on to part 2, how it all works. The hydrogen atoms produced in the Krebs cycle, which we talked about last tutorial, with the coenzyme NAD, which is attached to the crystal of the mitochondria. So the crystal would be about here somewhere on the black parts. The reduced NAD donates the electrons of the hydrogen atoms they are carrying to the first of the series of 3 electron carriers in the electron transport chain. Their hydrogen ions remain in solution in the mitochondrial matrix. The electrons released provide energy for the first proton pump. The energy is then used to pump protons into the intermembrane space, which is this part here, the peachy part. The electrons pass along the chain of carrier molecules, providing energy for the pump in turn. The protons then accumulate into the intermembrane space, as the inner membrane is impermeable to protons. The concentration in the intermembrane space becomes higher than the matrix, so a concentration gradient is formed. So basically, a concentration gradient in a nutshell is when I have a certain concentration here, and I have a higher concentration on this side of my membrane, and the gradient is favored more towards the areas of high concentration. So it'd be low to high in this case, but more on concentration gradients when I get round to a tutorial on it. So in the membranes are special protein channels. Each channel is associated with each channel in the enzyme ATP synthetase, or I've got it here in the textbook as ATP synthase. So depends which way you want to remember it, but ATP synthase and ATP synthetase give you marks. Protons then diffuse back into the matrix through these channels, and they do so. Their electrical potential energy is used to produce ATP. So electrical potential energy is another word for saying volts. Yes, electricity is all about electrical potential energy stored chemically. So really, this is one big battery, in a sense. And at the end of the chain, the electrons combine with these protons and oxygen to form water. So, if we zoom in to our diagram here, H plus ions there, and the membrane is impermeable to H plus ions, so the concentration gradient is always going to stay like that. So then reduced NAD goes through proton pump number 1, releases an electron. The electron carrier carries it to pump number 2, and then pump number 3, and the H plus ions get sucked up through the pump, and 4H plus 4E minus plus 1 oxygen equals 2 H2O. And then the enzyme ATP synthetase, then along with a phosphate and ADP and some energy, all make ATP. Now all of those ingredients are in the pink part, and the H plus ions are in the green part here. So that's the electron transport chain. Now, quick summary on the mitochondria, and a quick study point for you to appreciate. The Krebs cycle releases hydrogen atoms in which they can be used in oxidative phosphorylation to provide energy to make ATP. So that's a good point to make. So now finally we move on to part 3 and our final component, if oxygen is available. So oxygen is preferred as the final electron acceptor. Sounds like a very big role to have in the electron transport chain. Oxygen is essential in the process of the removal of protons and electrons. Without it, the protons and electrons will accumulate and cause a backup along the chain and bring the process to a halt. By a backup, they mean a backup of H plus ions and floating spare electrons, really. And the protons move down the concentration gradient through the ATP synthase. The energy released causes the rotor and stalk of the ATP synthase to rotate. So once chemical energy is given, then the synthase enzyme keeps rotating and rotating, and the rotation is converted into chemical energy as the phosphate is added to ADP to form ATP in the catalytic head, which is this funny thing here. FAD replaces NAD in the carrier system at the point in the Krebs cycle where 2 molecules of ATP are produced rather than 3 molecules of ATP. FAD passes the hydrogen atoms directly into the second pump. In other words, the carrier system involved in NAD has 3 pumps, whereas FAD carrier system has 2 pumps. So therefore the yield of ATP is therefore different. So NAD is probably preferred if you want more energy. So, quick summary of what we did. We found out what the electron transport chain was, and we introduced ourselves to chemio-osmotic theory. We reminded ourselves of oxidative phosphorylation and what a coenzyme was. We also introduced ourselves to the process of the electron transport chain and how all the electrons, along with reduced NAD and FAD, combine to make 2 molecules of water and a little bit of H plus ions, some phosphate, ADP and energy to make ATP, as well as that. This is the main goal of the electron transport chain. So all of these steps, you might want to write them on revision notes or watch my video again if you're that kind of person. And as for the mitochondria diagram, it's nice to take into account that the mitochondria looks like this. It's also recommended that you look at a chloroplast as well, as I will be talking about photosynthesis, and the electron transport chain will come up again. We finally talked about the final electron acceptor, or oxygen. So in the presence of oxygen, even more ATP will be made with our special enzyme here.